Genetically Modified Foods by Zohar Hassan, Ashani Patel, and Stacey Brennan. Introduction. Okay, so what are genetically modified foods? Well, let's start with a little background. Lately, there's a lot of people on the planet, which means we need a lot more food to feed everybody. How do we feed the masses with urban settings growing each and every day? Well, scientists think they have the solution. They think that genetically modified foods can help. A lot of people are like, you can't do this because things that come out of a laboratory can also, as beneficial as they can be, can cause super weeds and super bugs, and they can just cause a whole series of problems. So this argument can go back and forth because then scientists say that this can help the environment and this can help preserve land, and again, like I said, it can go back and forth. So what we did during this lab was discuss genetically modified foods, and we tested one of our foods that we eat on a regular basis. In this case, we chose Tostito chips, and we extracted its DNA, gave it a polymerase chain reaction to speed up and amplify its DNA, and then we, after setting up our controls, we put it through gel electrophoresis, and then we checked to see if it was genetically modified or not. This experiment was actually broken down into 3 days. So the first day was pretty much the setup of our sample. So first we weighed up our gram of Tostito chips. There's our gram. Hi, gram. And then we grinded it for a good 7 minutes until we were able to pipette it. And what we grinded it with was, this was a mesh of water and Tostitos. Then we pipetted 50 micrograms into the non-GMO and the test tube. So what we're going to see is a picture. Our test is our Tostito, while our non-GMO is actually oatmeal, which we already know for a fact is not genetically modified. So then we shook them to the left, and we shook them to the right, and we shook them from side to side, and then we put them in a water bath that was at 95 degrees Celsius for about 5 minutes. Actually, I think it was exactly 5 minutes. And even though that does not say 95, it was 95. And then we centrifuged the tubes. And the purpose of centrifuging was to really get separate layers to separate the layers, so the lighter material would float to the bottom and the heavier material would float down to the bottom, and the lighter material would float to the top, and the DNA would separate itself. And then we refrigerated them. And thus that was the end of day 1. So now we have day 2. So day 2 was really about setting up our experimental controls, and those are our positive and our negative controls. So what we had were 6 small test tubes. They were actually puny little things. And in test tubes 1 and 2, we had our oatmeal. And this served as our negative control for genetically modified foods. So we used this to make sure that within the following tubes, we could make sure that we had extracted the DNA correctly and to prove that our conclusion was correct. In tubes 3 and 4, we took our sketchy substance, which was in this case Tostitos, the mush Tostitos, and we don't know whether it's a positive or negative control, so they're just sketchy at the moment. Then we had tubes 5 and 6, in which we had a positive control, which means that the substance that was in these tubes was actually genetically modified. So these tubes, we added, in tubes 1, 3, and 5, we added plant master mix, which would amplify the primer found in plant DNA and would allow for it to replicate several times, so we would have more DNA to work with. Plant DNA, that is. Then in test tubes 2, 4, and 6, we added GMO master mix, which essentially did the exact same thing that the plant master mix did. It would find the primer for the genetically modified food and amplify it in order to have a greater number of that DNA. So that's us pipetting away, and sadly we have no background music, so bum ba-dum bum bum ba-dum bum ba-dum bum bum ba-dum bum bum ba-dum bum 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 bum. So, what we just saw was we were just adding the plant master mix and the GMO into the separate vials, and that's what we did for test tubes 1 through 6. Now we are at day 3, and what we did day 3 was actually test and see the results of whether we had genetically modified foods. So first we set up our gel electrophoresis. Sorry, my mom walked in the room. So the first thing we did was set up our gel electrophoresis, which is basically making our agarose gel, installing the comb to make sure that we had the wells, putting in the forts, as I like to call them, and excuse me. So we diluted our stock solution, and then what we did was we heated it, and then we put our PCR tubes into a thermal cycler, and then we put them into a microtubular adapter. And that's our cycler, our thermal cycler, which can raise and drop its temperatures very easily. And then we pulse spin the tubes for about 3 seconds, and then we loaded the samples into our wells for our gel electrophoresis. Once everything had been set up, our agarose gel had the well, so we pulled out the comb, and in our first well we put in the molecular weight ruler. In our second well we put in samples 1 and samples 2, which were actually our positive controls. In wells 4 and 5 we put in our sketchy substance, our tostitos, and in wells 6 and 7 we put our negative controls, which was our oatmeal. And there we are. So basically what this video is showing is us inserting the DNA into the wells. Now, gel electrophoresis is used for this because the DNA, because it has a negative charge, it will be pulled towards the positive charge, and this will pull the DNA, and based on size, what it will do is it will transfer. Now, plant DNA has 450, the plant primer for plant DNA has 455 base pairs, so that will go farther, yes. And then our genetically modified food will have 255 base pairs, and this will be evident as we put it through the gel electrophoresis charge and then stain it. So while it's in the wells, because our DNA is actually colorless, we had to stain it with Fast Blast, and we actually used orange. So add 10 microliters of orange leading dye into each PCR tube and mix. So that's what we did, and that's how we got our orange substances. And here is our DNA after we have put it through the gel electrophoresis. As we can see, it's moved down. However, they all look pretty similar. We can't see the traces of the DNA. until we take a couple more steps, which are the steps that follow are adding a chemical that will stain our DNA, and we put it in a baggie, and what that chemical did is it stained our DNA, and after balancing for a little while, we put it over a UV light. And now what we're looking at here is our gel electrophoresis samples under a UV light. So this is our actual DNA is what we're looking at. Now you'll see here in well number 1, this is our negative control for genetically modified foods. So this one had plant master mix, as you can see down here, and we did in fact find our DNA primer. However, this one had genetically modified master mix, and we can see that there's nothing here, and that's simply because oatmeal hasn't been genetically modified. It's a negative control. Here we find our sketchy substances and our tostitos, and for the plant master mix we find that there was the DNA primer. Oh, I'm sorry, this completely just shifted on me. We find that it did have the plant master mix, the plant DNA, so we know that we definitely extracted the DNA. But now if we look here, we find a faint line as to where the GMO master mix was added, and that tells us that yes, our food, although it may not be too prevalent, has been genetically modified. This is a positive result because we can see it lines up here with our positive result. And our 5 and our 6, okay, so this was our plant master mix for our positive control. This just tells us that there was DNA in our positive control for genetically modified foods. And then in well number 6, we'll see that the DNA has traveled far down enough and is equivalent to where our genetically modified tostitos are. This just tells us that yes, the food has been genetically modified. Here, a DNA marker is set up to tell us how far each of this DNA traveled. Now, if we'll take a look, 1, 3, and 5 are all plant DNA, and because they are larger molecules, they didn't transfer as far. However, the genetically modified foods, because they have smaller base pairs and smaller primers, they've traveled a bit farther, and they're actually 455 base pairs long. So our conclusion: sample 1 and sample 2, we already knew that they weren't genetically modified because they were our control. Samples 5 and 6, we already knew that they were genetically modified. However, samples 3 and 4 were a sketchy substance, our tostitos, and we didn't know if they were genetically modified. However, after staining them and after putting them through gel electrophoresis and staining them, we found that they do, in fact, have been, that was such bad grammar, they have been genetically modified. So we conclude that our Tostito chips have been tampered with. Okay, well, thanks a whole bunch, and I hope you enjoyed it. I think it was sort of laid back, but I hope that every point got across, and I hope you have a great night. Thanks. Bye.